Hello everyone, welcome to the ITLS Academy. Today we are going to perform an experiment that is your purification of the protein. And here we purify the protein by the method is dialysis membrane. Okay, so before starting the experiment, we have to know that what is this protein means. So simply a protein is what? Protein is a polymers of hundreds of amino acids. And these amino acids are linked together by a bond that is called a peptide bond. And proteins are present in a different structure, that is your primary, tertiary, secondary, quaternary. These are what? These are the structure of the protein. Now, before starting the experiment, we have to know that why we perform this purification of the protein. What is the aim of the purification of the protein, or you can say that what is the requirement of the purification of the protein? So, here four requirement of the purification of the protein. The first one is that we have to study the structure and the function of the protein that we desire. And the second thing is that we have to study the protein regulation and the protein interaction. Okay, the first one is that we have to identify the structure and the function of the protein that we desire. And the second is that we have to study the protein regulation and the protein interaction, and also it is used for the production of the antibodies. That's why the purification of the protein is very important. And the last point is that these proteins are used in a acid, so that's why we require the purification of the protein. Here the purification of the protein are done by a membrane that is called a dialysis membrane. You can see that this is what? This is a dialysis membrane. So basically, what does this dialysis mean? Simply, the dialysis is what? Dialysis is the separation of the larger molecules from the smaller molecules. And these smaller molecules are nothing. They are simply impurities and a salt which are present in a mixture of that crude enzyme or a salt precipitated enzyme. That is our sample. And this separation take place through a membrane that is called a semi-permeable membrane. Dialysis are based on a principle of diffusion, which simply means that the movement of the molecules from the higher concentration towards the lower concentration, and the membrane that we use here, that is a dialysis membrane, like your semi-permeable membrane. So, this dialysis membrane are made up of a cellulose acetate, and they have a very smaller pore size. Okay, due to this smaller pore size, it allows only transfer of the smaller molecules from the inside towards the outside the buffer. Buffer means that at which this dialysis membrane are kept. So basic is this only. What we do, we pour the crude enzyme inside the dialysis membrane, and then we pour this dialysis membrane inside the buffer and kept it at 4 degrees Celsius for overnight. And then this, due to the smaller pore size, the movement of the molecules take place from the higher concentration towards the lower concentration, and that's how the separation occurs. So for the dialysis of the protein, we require first dialysis membrane and sodium bicarbonate. Sodium bicarbonate is required for the activation of the protein molecules. Then we require a potassium phosphate monobasic and potassium phosphate monobasic and dibasic. Then with the help of these monobasic and dibasic, we prepare a potassium phosphate buffer, and also we required a crude enzyme and distilled water. Okay, so the instrument that we required is simply a weighing balance and a pH meter. pH meter is required to maintain the pH of the potassium phosphate buffer. So first of all, we have to activate this dialysis membrane because without activation, the experiment are not possible. So for the activation of this dialysis membrane, we require to dip this dialysis membrane inside the distilled water, and this distilled water is at 100 degrees Celsius, means that boiling distilled water for a 10 minutes. Then after 10 minutes, what we do, we pour this dialysis membrane inside the 2% of the sodium bicarbonate and again boil it for a 10 minutes. Then again after 10 minutes, what we do, we remove this dialysis membrane from the boiling sodium bicarbonate. Then again we boil it at distilled water for a 10 minutes. This is how we activate this dialysis membrane. So around 30 minutes is required for the activation of the dialysis membrane. So first of all, what we are going to do, we pour this dialysis membrane at 100 degrees Celsius. So just when the water start boiling, we pour it. Apart from it, we have to prepare our 2% of this sodium bicarbonate solution. So we prepared 2% of sodium bicarbonate solution for 200 ml. Okay, so for 200 ml, we required around 4 gram of this sodium bicarbonate and 200 ml of distilled water. So this is our 110 distilled water that we are going to prepare. So first of all, we take a 100 ml of distilled water. Now we weigh 4 gram of sodium bicarbonate. We have to tare it. Just 1.92. Now it is 4 gram. Now dissolve this 4 gram, first of all 100 ml of distilled water. When it is completely dissolved, then the remaining 100 ml of distilled water will be added. Until now this will be boiled. Now again we have to add a remaining 100 ml of distilled water. This is your 100 ml. So 2% of sodium bicarbonate is ready. Still, it is not boiled. So we prepare monobasic of this potassium phosphate. So for this is your monobasic. For monobasic, what we have to do, first of all, we take a 34.02 gram and dissolve into a 250 ml of distilled water. Now it is boiled. So what we have to do, we kept this dialysis membrane inside the boiling water for a 10 minutes. Then after 10 minutes, we pour this dialysis membrane inside the 2% of the sodium bicarbonate, and then again after 10 minutes, what we do, we again boil it for a distilled water. So it takes around 30 minutes for the activation of this dialysis membrane. Then after that, we prepare that buffer. So first of all, we activate it. Now our this dialysis membrane are activated. See, after 30 minutes, 10 minutes in a boiling distilled water, 10 minutes in a 2% of sodium bicarbonate, and again 10 minutes in boiling distilled water. After that, it is, see, it is activated. Now we have to prepare a 50 millimolar of potassium phosphate buffer. For this, we required a monobasic of potassium phosphate and a dibasic of potassium phosphate. So, first of all, we are going to prepare a monobasic of this potassium phosphate. For this, we required a 34.02 gram of monobasic potassium phosphate, and it will be dissolved into a 250 ml of distilled water. Now what we are going to do, first of all, we take a half of distilled water, like 110 ml of distilled water. Now I am going to weigh a 32 point 32 point potassium phosphate mono basic. This is 10 gram. Now dissolve this 10 gram into distilled water. Again take 10 gram. Now at last we take a 12 gram. Then it will become 32 point. So this value is calculated for a 50 millimolar of potassium phosphate buffer. And how we calculated for this, you can see the solution preparation video of our It's already uploaded in our account. You can go and see the video. Now we have to dissolve it. Say this is your stock solution because the concentration of the monobasic and diabasic is very high. Then with the help of this stock solution, we prepare our working solution that is called a potassium phosphate buffer. This is your monobasic. Now I am going to prepare a second buffer that is your diabasic. So for diabasic, we required a 43.54 gram and it will be dissolved into a 250 ml of distilled water. First of all, take a 250 ml of distilled water. Add 120 ml into it. Now we have to take a weight of diabasic. Now I am going to dissolve a 43.54 diabasic into 250 ml of distilled water. So this is 10 gram. First of all, I am going to dissolve 10 gram into distilled water, then again 10, and again 30. Now the last 10 gram I am going to take. Now I'm going to add this diabasic into a distilled water. Okay. Now mix it properly so that it will completely dissolve into a distilled water. When it is completely dissolved, the remaining distilled water will be added into a flask. Now our two solution, that is your monobasic and the diabasic, are ready. Now with the help of this, what we are going to do, we are going to prepare our potassium phosphate buffer. So for this, we take a 5 ml of monobasic and 5 ml of the diabasic into this. Then we maintain its pH 7, and then after that we make a volume 100 ml. So first of all, I am going to take a 5 ml of monobasic. 1, 2, 3, 4, and 5. Then change the pipette and change the tip, and again I am going to take a 5 ml of this diabasic. 1, 2, 3, 4, and 5. So 10 ml we take it. Now I am going to maintain its pH 7 with the help of the pH meter. It is slightly acidic, so we have to add only 2 to 3 drops of the NaOH into the buffer solution. I guess we don't need it because it's already Now, after tying this dialysis membrane from one end, I pour the 1 ml of this crude enzyme into it and then again tie it here. Now I kept it inside the beaker and add a 50 millimolar of potassium phosphate buffer so that it will completely dip inside it. Now this should be kept inside the refrigerator at 4 degrees Celsius for overnight. Then after incubation at 4 degrees Celsius, tomorrow we will keep it into the shaker and in a continuous shaker, and every 30 minutes we change this potassium phosphate buffer for 3 hours. Then after that we get our dialyzed protein. So we will see the result tomorrow. Now I am going to keep it inside the refrigerator for overnight. So after the overnight incubation at 4 degrees Celsius, we are going to put this beaker into a rotary shaker. Okay, now what we do, we change this potassium phosphate buffer every 30 minutes, and we do this for a 3 hours interval. Now I am going to put this conical flask into the shaker and start the shaker, and this should be continue still, means that it should be continue rotate inside the on the shaker. And every 30 minutes at 3 hours interval, I am change this buffer with the fresh buffer, potassium phosphate buffer, and after 3 hours we get our dialyzed protein. So we will meet after 3 hours. After 3 hours at every 30 minutes interval, we get our this dialyzed protein. Now I am removing our dialyzed bag from the I am removing the dialysis bag from the buffer solution, and this protein are known as a dialyzed protein, and it is used in a further experiment like ion exchange chromatography. And before performing the ion exchange chromatography, we have to centrifuge it. Okay, the reason behind the centrifugation is only that if there are any impurities, might be, but it is not possible that the impurities are present in this dialyzed bag, because it is removed due to the small pore size. But if any impurities are present, for the precaution purpose, we do a centrifugation, and then we get our dialyzed protein, and it is ready to use for a other experiment, that is your ion exchange experiment. So this is all about the purification of the protein by the method of dialysis. I hope you will understand, but if you have any queries related to this topic, you can ask me in a comment box, or for a further detail, you can follow the ITLS Academy on different platforms, that is Instagram, Facebook, Twitter, YouTube, WhatsApp and LinkedIn. And subscribe our channel ITLS Academy on YouTube, and visit our website www.itlsacademy.com. Thank you for watching. Please like, share and subscribe our channel.